Joining us now from CARR Biosystems is Product Manager Colleen O'Malley. Welcome, Colleen.
Thanks, Perry, for the introduction. My name is Colleen O'Malley, and today I'll be talking about cell handling across diverse cell types with a few specific examples with iPSCs, MSCs, and NK cells. A little bit of background about who CARR is. CARR was originally founded in 1993 and has since been rebranded as a part of the Berry-Waymiller Group. We have a team of about 100 members, and we have US manufacturing. And our goal is to provide enabling technology for globally licensed products. In terms of modalities that we're supporting, today we'll speak mostly to cell therapy, and we support therapy developers with programs across the pipeline, but we do also play in gene therapies, mAbs, vaccines, and cell agriculture. Today we'll be talking through the Unifuge system, which is our single-use centrifuge, and wanted to break it down into the three main components. We have the equipment, which is that hardware element. It comes in various sizes and has straightforward setup. There's the single-use module that comes sterile, gamma irradiated with the bowl and some tubing set so that it can be aseptically connected to those upstream and downstream process steps using either tube welding or aseptic connectors. And then lastly, we have the automation, which is that software side. It has PLC-based control, push button recipes, and real-time monitoring with options to have 21 CFR Part 11 compliance or SCADA and DCS integration if you are looking for setup within those GMP environments. In terms of the Unifuge family, things got a little askew here, but you can see the family. It will scale from bench to commercial. So we have the Unifuge Mini, which is our smallest piece of equipment. We see that used for both autologous and allogeneic cell therapies. And then for the Unifuge Pilot and U2K, those are scale-up options for our allogeneic cell therapy customers. And then in terms of the modules, the Unifuge Micro and the Unifuge Mini module both fit within the Unifuge Mini system. And the choice between those two is something our team can advise dependent on your working volumes, the cell count, and the target cell density you're looking for as you move into your downstream processing. And then the Unifuge Pilot and U2K fit within those respective pieces of equipment. In terms of how the Unifuge is used, there's a few typical use cases being harvest, wash, buffer media exchange, concentration, and even pre-formulation steps being that exchange into cryopreservation buffers. A little bit on how the Unifuge works. So like I mentioned, this is a tubular bowl centrifuge. So the cell suspension is loaded through that central feed tube. The bowl will begin to spin, and then the cells will gently accelerate outward along that bowl wall, separating through G-force. And then there's an option for a buffer wash and exchange step so that spent media can be removed and the buffer washes away those residual impurities. And then you can harvest those viable cells for downstream processing. I think the fundamental part of how the Unifuge works is it's giving that gentle processing, so it's low shear, and this is where we really are able to preserve the viability of the cells and achieve higher recovery. So I have an example cell therapy workflow here, and there are going to be nuances for every process and cell type. But what I want to highlight today is across the workflow, there's always going to be cell handling steps, media exchange, concentration, from upstream at cell selection all the way through to cryopreservation. And I want to pose the question, why does cell handling matter? First off, it's a source of variability. As we can see, it's repeated across almost every step, which compounds the impact, and it's a cause for unwanted aggregation. With this in mind, I want to talk through a few specific cell types now, being iPSCs, MSCs, and NK cells. So going to iPSCs, and I'll orient us a little bit with this slide, but want to talk through kind of what we're typically seeing, the workflow that we're usually working within, some of the handling challenges in practice, and then how the Unifuge can help control. So iPSCs are extremely shear sensitive and morphology dependent cell type. The workflow has a lot of repeated media exchange steps across multi-day long processes. So these media exchange steps occur from seed iPSCs at the single cell state as they expand into aggregates, transition into intermediate cells, and then ultimately differentiate into that target cell type. Some of the challenges we see from cell therapy developers and manufacturers is morphology changes. Those can occur at the single cell or aggregate level, and that may look like cell damage or even cell lysis, or it may be causing unwanted cell clumping. There can be a loss of pluripotency, meaning that differentiation capacity into the diverse target cell lines, and then variable media exchange being that it can impact the expansion capacity and cause inconsistent differentiation outcomes, meaning how well can it get to that target cell and when does differentiation occur, which ultimately is causing genetic instability of the cell type. Where the Unifuge is able to come and control some of these challenges is independent of single cell or aggregate size, we can handle both. This is due to the tunable critical process parameters of the system being flow rate and G-force that creates that low shear handling environment, preserving the morphology. Due to this and due to the consistency of the media exchange, we're able to deliver reproducible differentiation outcomes. And I want to dive into this a little bit more. I have a recent case study here with a cell therapy developer, and this is comparing to their incumbent setup of gravity sedimentation. This is a manual, tried and true, super gentle approach, but just simply not an option for commercializing as they look to scale up. So what we looked at here in comparison was the midpoint differentiation markers, and we were actually seeing that the Unifuge Micro was able to exceed that control, indicating that that gentle low shear handling was matching and exceeding expectations. That was similarly what we saw at the endpoint differentiation markers, and this case study is available if there's anyone interested in learning more. Next, I want to talk through MSCs. So typically what we're hearing frustration around is some of the harvest variability. So from expansion, these MSCs are going through harvest, wash, and concentration steps. Some of the challenges that we see is MSCs are known to be sticky, which can cause unwanted aggregation. Dependent on the source material, whether it's umbilical cord or bone marrow derived, there may be differences in how these cells behave. And then similarly with culture, making that transition from adherent to suspension culture, there can be some behavioral differences. And then one of the biggest things we're seeing right now is frustration and challenge around limited scalability. Folks are looking to get to their next clinical milestones and make sure they're commercially ready. And a lot of people right now are trying to track towards that 50 to 80 liter scale, with some even looking to 200 liter scale. And their current setup just isn't something that can grow with them. So where the Unifuge comes in is we do have strategies to disperse those MSC clumps without damaging the cells themselves, and that increases recovery. The high recovery is also a function of decreased processing time, which minimizes the cell settling. And then the recipe-driven automation, that's something our team can come in and guide recipe development as you do adjust between source materials or culture format, or even if you're looking to scale up from the Unifuge Micro to the Mini to the Pilot and beyond. I'm going to linger a bit more on the high recovery once again and wanted to show another case study with a cell therapy collaborator. And this we were comparing to their incumbent spinning membrane filtration. The recovery they saw was typically 60 to 75%. And these three trials here were the first 3 trial runs completed. So right out of the gate, we were able to hit greater than 90% recovery for all the runs. And that's before any further process optimization. Additionally, we were able to cut their processing time in half for this specific example. The last cell type I want to talk about today is with NK cells. So these cells have issues with adhesion, especially looking at wash and concentration steps. These activated NK cells become sticky. The increased processing time can also lead to more adhesion-related issues, as well as viability losses. And then inefficient washing tends to lead to inconsistent reagent removal or washout of cytokines. Where the Unifuge comes in, you can adjust those process parameters to disperse the NK clumps. The gentle and efficient handling preserves the cell health as we think about phenotype and function, and once again, getting that high recovery with precise washing. And precise washing, and what we're thinking about, washing efficiency is really a function of wash volume, the number of washes, the type of wash media, and we can reach up to 99% wash efficiency, but it's more about getting the consistency of what your target wash efficiency is so you can lock it in, especially as you prepare for maybe a cryopreservation or pre-formulation step. So we talked through a few examples, but typically seeing greater than 95% recovery, about 98% viability. We've worked with 30 plus different cell types and other biologic materials. So if you're not seeing what you're working with today mentioned, doesn't mean we haven't done it. And we'd love to talk more about what you all are working on. And the key takeaway I want to leave you all with today is cell handling impacts health, recovery, and quality. Gentle, low shear processing ensures high viability for the cells. The Unifuge delivers consistent quality, so it's reproducible across runs. And with this automated system, you're having efficient operations with near walkaway recipes within a closed system, reducing your risk. And then you're able to scale up and consistently hit your recovery targets from one system to the next. So I'll kind of close out here. Our team is out and about globally. We run over 200 demos annually, the majority of which are with cell therapy folks. So we are happy to come to your lab and try this out with your cells or talk through what your challenges may be and be able to share some of the learnings we have from our experience. With that, thank you for your time, and I'd be happy to take any questions, Perry.
Very interesting, Colleen. Great presentation. Can you address what kind of volumes can the Unifuge process, and can it be operated as a closed system?
Yeah, so the typical process volumes that we're looking for, depending on what your discharge, what you're looking for that downstream processing, is really dependent on the cell count. But typically we're seeing somewhere around the 100 milliliter mark as the minimum, and then we can scale up quite large for the U2K, kind of beyond where most cell therapies are at today, getting up to thousands of liters. So that's our typical range. And then in terms of it being a closed system, like I mentioned with the single-use module portion, it can be aseptically connected to your upstream bioreactor step and downstream whatever's next, using tube welding or aseptic connectors. So all of it will remain a closed system.
So in workflows with repeated media exchange, how do you balance speed versus gentleness during handling?
Yeah, I think these two really go hand in hand. So I think a lot of time when folks are early on in their lab and kind of development journey, they're just focusing on getting something done, and they're going to be turning to what's within their lab already. So oftentimes they're using some of these manual methods like gravity sedimentation, and that definitely hits the gentle mark and it will keep it pretty fast from a lower volume. But I think oftentimes teams are challenged once they're hitting scale up to meet that same processing time requirements and obviously ensure that there are no consequences to having longer processing time. For example, those cells are getting taken outside of their ideal environment, so it can have some impacts there as well.
Yeah. What have you found that, or have you found that cell types or cells respond differently to the same handling steps?
Definitely. I think with iPSCs, that's a very shear sensitive cell type, and we're often seeing it used both in the single cell and in aggregate. So those respond very differently in comparison to another cell type. For example, MSCs, that's going to be sticky and have differences there. So our team has great experience out in the field and has a lot of intuition and years of experience around this. So they're able to kind of come in with the best recommendation of a starting point, and then we can always optimize from there.
Yeah, great job. Just a very interesting topic, handling of cells. So critical, so important. Thanks for bringing that to the event. Colleen O'Malley from CARR Biosystems.